or are you all absent?
We are ready.
We're ready. All right, excellent.
All right.
So we will get started. So welcome back to the second half.
I will check the time.
I think we're... So the second part is going to cover some of the same territory from the previous session, but it shouldn't all be mostly new. It's given me a chance to, again, look ahead. So I have been able to present this project a few times at Novia, and that's been really, really nice to be able to do that. And generally speaking, I've had to, or felt like I had to do what Joel was doing in the morning for about 45 minutes, and then really quickly talk about our project at the end, because so many of these concepts are rather new. So I'm really grateful that Joel was able to join us this morning to talk about these things. And then also for Ricard to share his journey, and also this very important thing about practicalities. How is this actually going to work? Spoiler alert, I don't know, but we're going to talk about it here in this section. So, okay, this is a little bit of lag, but now it's back. Someone's seen some of these things before. So the topics for today: adaptive nutrient management. So that's what we left off with. We couldn't get into it, but I snuck it in here. What are we aiming for at Lill-Nägels? What are we trying to go for? And then temperate, successional agroforestry, all in 1 hour. So bear with me. We're going to try to get through this and see where it goes to the question and answers. So these pictures here, we have kind of 4 topics, and hopefully there's something that's a bit interesting here, not just a little plan on paper. Oh, right, question and answer. Always have to have that. Adaptive nutrient management. Now, so I understand that farmers already, in some sense, practice adaptive nutrient management, because you just don't do the same thing every year. It's probably illegal to do the same thing every single year. But what we're talking about here is about adaptive nutrient management during the growing season, and with data from the plants to support those decisions. So let's talk about this a little bit. This is all available on the website, so I'm going to go rather quickly, just so that if you're unfamiliar with what we're trying to do, why, where, and when are we testing the soil at this project. This schematic is a little bit out of date. You can see it doesn't have all the same things, but in general, because even though the details of the design have changed a bit since this was published on the website, obviously the soil hasn't changed that much. So last year we had enough funding that we could look at the soil in 2 places with using conventional and near-infrared testing. So I divided the site into where we have a moister, deep soil that's mostly clay loam, and where we have a more sandy loam that's very shallow, dry, drought-prone, and we use those as the baseline then for the trajectories that what we're going to talk about in the next few minutes go from. So that's kind of more or less where they divide in the site, and then we are going to be testing in a few different locations. But the question that we have, why, is asking, will divergent goals, the plant communities associated with those goals, so we have different goals, so we need different groups of organisms to hopefully move towards that, and then the subsequent adaptive management of those communities alter measurable parameters. Now, again, correlation, causation, our project can't, say anything, but we can generate preliminary data that we can incorporate into our project's management. So we're not going to claim anything about how these things actually change specific measurable traits in the soil, but we can say that they have changed during this time frame, and that's fair enough. So that's the main question. So we want to regularly monitor the soil in at least 4 zones. We're able to do 4, which is pretty cool. So we have the tree lines we've talked about. So one of those is the shallow sand loam soil, and then the deeper clay loam soil. In the earlier iterations, I'd actually had different polycultures according to the different places on the tree lines, but it got weird in terms of planning. So I decided, I'd actually kind of rather see how the same plant community fares with irrigation in those 2 soil places. So we're going to test that. And then we're going to check the alleys, but we're going to stay within the deeper soil, because actually the sections of the alleys that have shallow, poor soil, they have a lot of stones as well. You can't really cultivate them. Not as interesting. we have a lot more clay loam soil on Ricard's farm than we do this other sandy one. So we're going to go with the bulk area. So this average type, and then the margins, right? So we're going to, again, test that deeper clay loam soil. We're not going to really look at the sandy stuff except for underneath the tree lines. The samples are collected in late May, late August, and late October. And the point here is that if the idea is to see how the communities of organisms are altering the soil, you actually got to wait for them to do anything. Right? So you could test it as soon as the snow melts, but the plant community there hasn't barely woken up yet. So they're not really altering the soil that much. So if we wait till late May, then they have 4 to 6 weeks in some cases to actually do something where there'll be a change perhaps from the test from the previous October. Then late August gives time for the summer crops. If we introduce C4 plants into some of these cover crop mixtures as well, then that gives them some time to do a little bit of something. And then late October, because that's when photosynthesis basically shuts down. It's not true for all plants, but basically we're done at that point. And we also haven't like totally frozen out all the microbes yet. So we get that last little trace of something. And for me, it's really important to do that, to watch this over time. And clearly, 1 year is not enough time to say anything. We understand that, but you have to start collecting. You can't just make up data. So another issue here that I want to mention is that where we think we're going to see changes occur most rapidly. So the tree lines is where we think we'll see the most change the most quickly because we have that horse manure compost. A lot of that's going to oxidize, it's going to go to the atmosphere, but yes, there will be some deposition into that soil profile. We have put perennial plant communities there. They can absorb, the idea is that they're going to absorb any of those nutrients, build that into the system, just as Joel was talking about this morning, and then start cycling that. And we can help them even further because we have an irrigation system. So they have like the biggest leg up out of all these diverse communities are in the tree line. So we should see those not do this, take off, but we should generally see a trend. The alleys are actually going to be the slowest because we're trying to go from this grass back to an arable system growing annuals and some biennials. And so if you get your relay cropping and everything in really well, you can build soil with annuals. But it's not going to work as well as perennials. And then the margins are going to be somewhat in the middle. They kind of serve a bit as it's not a control, so don't hear me wrong, this is not that kind of project, but they're the most closely related to the pre-existing vegetation. So the pre-existing vegetation, mostly grass, mowed once a year. Well, what happens if we have these margins? We introduce biological diversity into them and still mostly manage them kind of the same way. Maybe that'll have an impact on how we think about funding these nature setbacks and these subsidies. Should we be able to regenerate the soil faster then if we reintroduce some diversity? So each one of these plant communities is playing a different role. How are we going to test them? Well, third-party testing is really important to us because I can't do it and you wouldn't want me to. So we send our samples off to Agroforestry. They had the packages that we were interested in doing in the same place. It's no use sending off samples to multiple labs if the same place can do the same thing. So they are looking at the conventional with reserves, it's their organic package, and the near infrared. So you can read a little bit of pros and cons to each of those on your own, but these are things that farmers are either really used to seeing, or at least they have the option to click it and buy it. It's already off the shelf. So this generates information that farmers are used to looking at a pretty good price point, especially for the conventional testing. And also introduces some cool stuff with the near infrared that the Baltic Sea Action Group, this carbon action, they were one of the main impetuses for the purchase of the machinery, I think, at Agroforestry to do some of the testing for the carbon group. So we're kind of piggybacking on the work of others, which is basically all I ever do because it's hard to come up with new ideas. Then lastly, we're also using Dr. Elaine Ingham's soil food web. And I put that in quotes, not because I don't think it's real, but it's just sort of her. Actually, it should have been here. It should have been soil food web in quotes. And this is actually being done by a gentleman known as Kimmo England from Uoma Earth Care. He's sitting here. So Kimmo lives in Vanta, which is very close to Espoo. And so if you're going to look at bacteria lifespan is, hours, minutes, I don't know, maybe some live for days, but it's very short. So I don't want to send my soil biological assessment where someone's actually going to look and count different groups of microbes and have it sit in Posty or whoever else for a few days and have those populations shift. Chemo can come in a few minutes to the site, take the samples the way that he wants to take the samples, that he needs to take them, bring them back to his lab that he has, and then he can, within the appropriate time frame, take a look at the soil. It's also the fact of the matter that there are very few people who are trained in this method to do this. So the proximity and the training is really important. And the idea about doing each zone 3 times a year, 3 different ways with third parties, 3, 3, 3, 3, 3, is that no one of these tools is perfect. We need a holistic view of what's going on in the soil, as you can see with these pros and cons. There's almost no useful information about carbon or biology coming from the first test. It's just so low resolution what you do get that it's not that great. So the near infrared complements that and adds the nitrogen. The Dr. Lane Ingham goes a step further because the near infrared tells you something about the microbes, but it's an inference based on carbon and nitrogen. No one's actually looking and counting, but it's interesting. So they complement, they contradict, which is also fun because contradictions is like, well, why are they contradicting? What's going on? And then lastly, this other one, that's not how much soil needs to be removed to look at anything underneath the microscope. This is where we put all of that into context. We take soil profiles just once a year because I'm not going to dig 4 soil profiles 3 times a year and rip up my tree roots. Once a year is going to give enough time frame that I can disturb the soil and do this. Otherwise, it's like, just digging pits in this field every few months. But this is just demonstrating a bit of the soil that we have there in the sandy. This is a pile of sand. So try to put all this into context because the numbers, the photographs, they're all really good, but none of those, of course, and they don't claim to, is going to tell you if there's a compaction layer. They're not going to tell you 60 centimeter rooting depth. You've got to dig 60 centimeters down if you want to see that. The lab tells you what the lab is capable of doing based on the parameters they say of how they test. So we want to take a step back and look at our whole system and think about, okay, where should we invest the next action in the system? Here's our good friend again, the pH chart. All right, so according to soil testing, here's one from the spring of this year. You don't need to know where they came from. You'll find that on the website later. But pH, calcium, and phosphorus are listed here. They use a very nice little system to kind of, each sample's a little bit different because it's based on the pH and CAC, whether or not they color you a full red circle or a half red circle. But we can see that our pHs are rather low, except this was the sandy one. It was kind of high. The calcium is low availability of calcium, and low availability, sorry, I had that backwards. This is the sand, and then this is the margin. I said you didn't need to know when I told you anyway. But you can see here that there's almost no phosphorus readily available. And I'll mention quickly that 10 in the fall was a 6. So 60% of it. So this is how fast some of these numbers can fluctuate. Because of course they don't hold your soil at the lab, test it, send it back to you, and then you put it in the ground, then you grow stuff, then you take it and send it back. It doesn't work that way, right? We all know this. So there's going to be a lot of variation, which is why we need a number of years to track these changes to see what's going on. But so even this, when it was a 6, then it wasn't going to be a nice green box. It was going to be more of our generally low. So
can I ask you, are you taking it from the same place?
we have the zones, and we take it from the same general area in the zone. So we don't go from like margin 1 at the southeast end of the field and then go all the way to margin 3 on the far side.
That's important.
So we are keeping the samples within the tree lines are from the same tree lines, for the same section more or less. Yeah, but as close as we can without just constantly scooping from the same spot. But we're trying to keep it pretty clustered. And Chemo and I contact each other because I send the conventional tests to the lab. And then he knows then exactly where he needs to also harvest the soil samples from. Harvest.
Just a comment to that the smart thing would be to take a GPS position from those samples. But if you are
Yeah, I mean, it's
even
within,
a couple of meters, because it can be very big differences.
Yeah, we're talking, and you have to take multiple samples and mix them together and everything like this. So we're doing it more or less a standard way, and we're trying to reduce, because we're not doing the whole 8,000 square meters in the whole big zones. We have like subsections of each zone to try to keep it more or less the same. Because we could cheat, actually, because the management changes in the tree lines, and you could say, well, this section is looking better now in the summer than this over here. And then you could try to pick up things. So we're trying to keep it, pretty standard.
The method would be good to describe in the.
Okay, I think it's on the website. Yeah. So we're just looking at these three for now because these are the three that I want to look at. There's a lot that's been tested. So the asterisk is that what we've already talked about is that there's a lot. Yeah. So that there's a lot going on in terms of the soil testing, but it's going to take years to really say anything. But one thing that I did think was pretty interesting from the near infrared was this dip in organic matter. So you can see everybody's starting from the same baselines from the summer 2022 when we had the first samples. And look at how the margins in the alleys dipped in the end of May. So those soils had been tilled and harrowed before the soil samples were taken, and we see that dip, and you don't see as strong a dip in the shallow or the deep soil tree lines because that disturbance had taken place already in July of the year before. I'm not saying that actually picked it up, but it's an interesting thing. But then watch how they climb again with this summer. So if it's photosynthesis driven, this change in soil organic matter, we're not claiming anything here, folks, but just trying to get you the general idea of what we're looking for. This would help explain why the spring samples were so poor. I mean, this is us in June. So you can see, certainly we lost some soil organic matter from that tillage and the heat and everything. But then this is actually taken the same day that we took those samples. Look how much photosynthesis is now taking place. So we're hoping that we can identify trends over a number of years if we can continue to have funding, or if it becomes so profitable that we can continue to self-fund it. We'll have to see. Where this starts to get interesting, though, is because it's only 1 year, is the Sap analysis. So Sap analysis kind of tells you what the plants are mobilizing from the soil. So it's one thing to have the soil report or even the biological, but you're not testing the plant. You're testing something else that as we saw from Joel's perspective, it's all one. As Ray Archuleta would even say, it's all one. Yes, it is, but each plant, because of those associations, has the ability or inability to capture nutrients from the soil. Same soil, different plants, different result. Then is there any correlation between what these different soil analyses suggest and what we can measure with other tools? Are we seeing something there? And Sap analysis, just very briefly, you take leaves, healthy leaves from the sample plants, send them to a laboratory, they squeeze out the juice, and then they analyze that juice 26 different ways, 26 different independent variables that you get back for each sample. And so that covers nitrogen, calcium, and the micronutrients, which aren't even on the tests that we have chosen to do. So things like boron. Boron's not on the near infrared or the other one, but it's a pretty important nutrient for photosynthesis. And moly, of course, isn't up there either. So what species did we end up testing this year? Again, remember there's a drought, so like with the garlic sometimes might only produce between 8 and 10 leaves before it's done for the whole season. So if you take one leaf from your garlic plant, you're taking at least 10% of its photosynthetic production capability of the whole season away. So you need a large population to sample from so you don't kill your crop. Right? I got to know what I have to do to nutrient the manage. It's like, I don't need nutrients, I need leaves. So when we had the drought that really wiped out a large part of our population because the irrigation wasn't there, we didn't get as much data as we'd like, but we did test the garlic. We did the dominant grass, the couch grass, this running grass, because it's spread throughout the field. we would be able to, it would be a commonality between the alleys and the margins. We knew that the tillage wouldn't kill it. We were hoping that the cover crop would suppress it, but we were hoping it would become an element in a diverse plant community rather than the, dominant species. So we tested that one. Rhubarb, which doesn't have parameters, so the lab can't tell you whether the numbers it gives you that it measured are good or bad based on their information, but they can still tell you what it is, which is really interesting. Because the rhubarb, as we've said before, is going to be a biomass plant, a support species, as well as a food plant. So what is it mobilizing from the soil? We can't talk about that today. It'd be a whole other webinar, really just talk about what we found from this. But there is a good blog on the Novialia blog that was written by Florentine, myself, about the sap analysis. He talks a little bit more detail. But the rhubarb definitely has a different profile than these other ones. And then we also tested the tillage radishes that were growing in one of the tree lines because we wanted to see how that differed from what was the garlic getting from the tree lines. Well, what is the radish? Because radish is non-mycorrhizal. All right, so it doesn't mean that it doesn't benefit from mycorrhizal fungi in the soil, but it's more of, it's not independent, but it's one of the weirdos because 90% of all plants associate, and then you have the brassicas who don't. So we're going to just quickly look at some of the results for the nitrogen, phosphorus, and calcium that we got this year looking at the grass. Because this is one that we tested all 4 alleys, alley 1, alley 2, alley 3, alley 4. Then we tested the same grass from the margins. So we also have a little bit more data as well. And again, remember, there's a drought. You saw the picture. There's nothing to take samples from, so there's some gaps here. We would have liked to do a lot more sampling, but you can only sample what grows. But the blue lines show you the optimal range. I'm not an expert on making graphs, so I had to do it this way. The low end is a little bit of 1,000 parts per million total nitrogen, not nitrate. They also give you that. They also tell you a bit more about the nitrogen, which is really nice. Really nice. Then above 2,000. So if you're within this range, they think that's optimum for grass. If you're above, you're in excess. If you're below, then you're deficient. The near-infrared test, which I didn't put on the previous slide, was showing that our nitrogen release capacity was less than optimum and actually low on some of the samples from last year. By spring, it had improved a bit, some differences. So we should have struggled a bit with nitrogen. But what we see here is that it wasn't until alley 4 anyway, at the end of the year, where it was even at the bottom of the optimum. And what I need to point something out here is that this field was put into grass in 2019. And when you put it into grass and you receive your subsidy, you're not allowed to do anything with fertilizer anymore. No nitrogen has been applied to this system, to the soil, since 2018, 2019. That's long enough for any nitrogen, especially inorganic nitrogen, to either have exited the system or become bound in the soil mineral matrix. I'm not claiming that we unlocked it from that matrix, but if you saw the color of that grass and everything as a farmer, you would think it needed nitrogen too. And that's what the soil test said, put on some nitrogen. Well, we didn't put on any nitrogen, except for on July 24th when we did that spray. And that's another story, we're not going to talk about that, but you can see that it did move the needle a little bit, or the needle moved during the time in which the plants were sprayed. That's maybe the right way to put it. So that's nitrogen, nothing added to the soil, plenty available. Also very few nitrogen fixers, folks. So it's not like this is clover and grass. This is just grass, and it's got enough nitrogen. Again, the grasses with the phosphorus, way in excess on some of these. So phosphorus, again, either exits the system or becomes so tightly bound to the soil, to put it simply, that the only way to get it out of the soil again is generally through phosphorus solubilizing bacteria, or in particular mycorrhizal fungi. And as Chemo can tell you, well, can you get to the resolution where you would see a mycorrhizal fungi hyphae in your soil samples? Not to put you on the spot.
No, not really, no.
No, but, well, let's just say that I'm quite certain that given the history of that site that we wouldn't have high mycorrhizal populations suddenly just transferring phosphorus. So phosphorus, again, it was low, remember? If it was low, you need to add it, it's in excess. Same thing again, same story. Calcium is the only one where it's kind of close. Calcium is kind of correlating so far with what we saw from the soil. it definitely starting to get towards the bottom of the optimum range, but even calcium was fine. I don't know what's going on. All I know is that we saw this again and again and again. The things that the soil samples said shouldn't be available were in excess when we tested the sap. Some people don't like sap analysis. They'll say it's bunk, we don't like it. Do a tissue test. As you saw from Joel this morning, you can argue about anything in agriculture. But I think that this is rather interesting, that we don't need to be applying those things. So then the question is to amend or not amend the soil. We're going to use these sap analyses to do a few different things, like do the seed treatments that he talked about. That's a great leverage point, is apply things to seeds, but also the initial foliar applications. We can already start to formulate what we think we need in the spring based on last year's data, because the soil hasn't really improved that much that we could, pretend that it's ready to supply these things. So as an example, let's look at garlic. So this is actually what the report would look like from garlic, from Nova Crop Control. You can see all the different parameters. You can see in the middle is the optimum, deficient, in excess, and then if the graph goes all the way, your bar goes all the way down to the graph, that means they had to chop it off somewhere. So it could actually continue to go on. So what did we identify from here? In the first foliar application, we can see calcium, boron, and molybdenum. David.
Molybdenum.
There we go. Molly or low. So we can already start to put this in here. The asterisks are just representing like how deficient was it. Like one asterisk a little bit just to give you an idea that, okay, these are what we need to start thinking about over the winter. How are we going to apply nutrients to the leaves in May so that we can already overcome, because this is based on that 12th of May sample. So we know that coming out of winter, the garlic was struggling with these, and our soil hasn't gotten that much better. So we're going to want to have that on hand. But then we also want to have, as you can see down here, things like zinc and potassium and copper and iron, which is supposed to be abundant at low soil pH, not in our case. It was tested with the sap analysis. We want to have these already available in the spring, so that after we apply the first foliar application, and then we wait a week and we take the samples from the garlic, we get the sample back about a week later, that then we immediately get that analysis and we start to combine things and go out as fast as we can to the field and apply again, wait a week and sample. We'll probably do it twice or maybe 3 times with the garlic next year. But we don't know, because as soon as you change something, then with the antagonisms and everything in the soil, there's a lot going on. You can't guarantee that it's a roadmap, but you just want to have these things available. I do want to say that we're not just like dumping nutrients. We are also applying these with chelators. So we're using our own compost extracts, seaweed extracts, could be fish hydrolysate. If we decide we want to add some protein, nitrogen into the situation. So we're not just applying straight nutrients. We are combining these things with carbon to help that microbial community. And this is how we want to tweak the above ground parts of the plant so that they photosynthesize better, more root exudates, and everything Joel talked about. That's like I just add that on it. And everything that Joel talked about in the morning. So that's how we won't necessarily directly interfere in the soil. The plants are going to choose what they do next. They're going to choose what they do next based on what they just got sprayed on, and that biology is going to respond. And then over time, this testing regime is supposed to, hopefully, discover some kind of signal. God, I hope I'm not out of
Can I
time.
ask, how are you deciding about the amounts of these nutrients? Because boron is poisonous.
Yeah, yeah.
In
Well, we
excess.
look. You have to first. You should always look at the recommended rates
Yeah.
for the whatever it is
For
that
the
you're
plants,
applying,
for
right?
garlic, yeah.
And then you have to also think, well, is there also boron in the seaweed? Which there is.
Yeah.
So then so the winter time after this seminar and after my final meeting, then I can actually think about it, because we have until you want everything ready in April. The first couple leaves come up. So we have to talk about it, So we'll see. Lower is better because we plan to spray again. Right. So what are we aiming for? This has got to be really quick. So here's just a nice photo, I think, anyway. Again, photosynthesis. We have alley, tree line, and a bit of margin in the top right corner. This is a whole system. Joel talked about this morning. Every component's got a role. So in our system, those margins, the percentage is depending on how many of these perimeter tree lines actually get put into play. They're a bit larger in our project. Again, because we're doing a novel thing in the alleys, so we want to make it simple when it comes to collecting yield data and performance of these novel systems in the alley. Just make these rectangles. So I do have my little plots. You know, it's kind of like my market garden, but blown up much bigger. So those are what we're doing here with habitat, water management. In some EU countries, you'll get paid per linear meter of these kind of systems to put in. How the subsidies work, we don't know. But mulch is something, it's a secondary mulch production for us. We can also harvest that. And then there's a question mark on the grazing. You never know. You know, one way to monetize a perennial cover crop is to graze that cover crop, once maybe in the initial year, and then as many times as the regrowth aroused in the second year, so that you have livestock on the land incorporating the livestock. So this would serve as a kind of a transit and buffer zone. The alleys make up almost half of the site, depends on that replan. It's enough. That's just under, an imperial acre, which is nice because that translates pretty easily to hectares as well. And that's enough land that you can start saying, okay, this isn't 10 wheat plants in a little trial plot. This is an 80-meter wide row that is, 6 meters wide. You're going to see it's performing or not. Then you can actually you could justify bringing the combine out, because we don't have one of these, researcher combines that's going to come out and go beep, 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 and measure every single seed. We're going to come out there and we're going to harvest it with that existing equipment. But this is going to be done with novel methods, and I put that in quotes because these aren't novel. This is the system that's most likely to work, these first two. People are doing diverse pasture kind of systems already, that we know that this works. Regenerative agriculture is already talking about growing broadacre things. But what we're going to do is move away from monocultures. So the closest we're going to get to a monoculture is probably planting a multi-variety mix. So if we plant spring wheat, we would try to find 3, maybe 4 cultivars of spring wheat that have complementary ripening times. So the seed companies like, why are you buying these competing seeds? But we're going to try to bring them together, because even just changing the varieties, even though it's still wheat, has been tested in research to bring benefits. So that's like the most simple we're going to go.
That's what medieval
It's called
period were growing many varieties of wheat, and the quality of the bread was much higher in nutrients. And that's been proven by finding thatch houses that still had bits of medieval thatch in it, and then analysing the species of wheat in it from einkorn, etc.
Right.
And then making bread out of the possible varieties that are available today, as close to the medieval varieties, and then finding that the quality and nutrient availability of that bread was so much better, and the crops were healthier. So
this it's not new. Some people are still practicing these things in Europe, and even combining oats and wheat and barley, and that's called a maslin. And there's been some recent study come out looking precisely what you just talked about. But of course, the end users, the big mills, don't want that. So this is an issue again that the EU is already recognizing that we got to take a look at. And the tree lines provide essential services for the alleys, then stacked with these cash crops. If we look at each of these different components as quickly as we can, a little bit beyond habitat and mulch, these are the goals for these areas here. We're improving that functionality. I've already kind of talked on this a bit. Secondary mulch production. So why would we bother to do this? The arrows are just pointing out these wet spots, and then standing water here in the third margin after a lot of rain. And so we also track, with observations, what's going on in the field. Margins and headlands tend to be less productive anyway. You hear this over and over again from farmers. So, you can try to force it to make a crop, but if you have a yield monitor, you'll be like, why am I bothering to grow? And that's why the EU is often saying, let's just go ahead and plant these margins. But our margins are a lot bigger, as I've said, than it would be because of this novel information production. So you would be able to generate lots of good information if you scale up the agroforestry. You have lots of plots doing many different things. We're doing many different things in one plot. So the margins are kind of a bit bigger, so we can do that. Yeah, they're wet. How are we going to accomplish this? Well, perennial non-woody plant communities. One thing that's different than what's generally talked about even in regenerative agriculture is that we're trying to reduce the grass and legumes. So grass and legumes is pretty much all we can ever think about in agriculture. Whereas we're discovering, when we look at the soil, we're seeing that there's a lot of free associating nitrogen-fixing bacteria. We can see in some studies, especially in this context of non-woody perennial communities, that you might even fix more total nitrogen with no legumes involved. Take the legumes away and the soil has more nitrogen. So we're going out on a limb. We're testing some of these ideas that are floating around. There's still going to be grass, there's still going to be legumes, but we're not turning this into a grass legume clover sward. Possible revenue streams, as I mentioned before, we've got these perimeter tree lines that I'm trying to squeeze in, but we got to get the combine out there and kind of ground truth it if it's going to make sense or not. And then, of course, as I said, this holding ground for animals. As you move them around and then you shoot them down the alley, they can still make use of the space. And then we have planted, as you see my foot over here, that's a ribwort plantain. We have a couple species of those that we put in from a biodiverse herbal ley that's being promoted in Ireland for commercial dairy farmers because they feel they have enough research now to show that what I just said, you can reduce your nitrogen use and improve the whole ecosystem by adding functional biodiversity. And then on top of all of that, these are medicinal for the animals. So not every animal is going to be, vet, they don't have to bring it to the vet, but you do see reduced vet bills because the diversity has compounds that influence their gut and influence their health. So this is also kind of preloading, also just in case we do have animals. Also, it's super affordable and you can just buy it. You don't have to think about it. Next, forays into crop associations. This is what the EU is calling intercrops and polycultures. I thought it was kind of funny. It sounds kind of like mafia crop associations. You heard of the association? Half of the researchers were like, I don't know what they mean by this. But yes, we talked about this a little bit already. Broadacre, novel techniques. This would be the primary mulch production. There's a question about mulch. We are producing mulch with the cover crops in the tree lines, but instead of making a new diagram, I thought I'd use the one I sent to Ricard last winter, demonstrating how you can use strip cropping and cover crops adjacent to the tree lines to provide access during the growing season so that you're not driving over your intercrop. You don't want to drive over wheat in August. It's not a good idea to get to your fruit tree stuck in the field. So considering that we have really poor soil, what we can do is have a strip of cover crop next to where the garlic is growing, especially in the first few years, give you access to that and the trees. Then in the next year, when the garlic moves to the other side, then you just swap. And you can do strip cropping in a variety of different ways, but we'll probably end up just actually squinching them together, not doing this kind of ribbon pattern so early. We'll probably have 6 meters of cover crops, 6 meters of cash crops, and then as the soil changes, then we reintroduce more cash crops. And this is what we foresee for the beginning. It's going to take a while, but that cover crop can also be cut. You can manage that growth curve. Remember what he was saying about the annuals and their root exudates. They reduce the root exudation and soil formation as they senesce and go to seed. So if you cut them at the right point, then some of them will die, but many of them will start to regrow from the biodiverse mix. When you cut them, you remove that residue so you don't smother your plants, you put it next to your trees, and then you have the cover crop growing again, so you keep riding the photosynthetic curve rather than just letting this go to seed. Annual-based plant communities are possible to restore soil. Cover cropping is not becoming a thing just because seed companies want to sell you more seeds, but because there's enough evidence that this works. So we're going to be doing that. And then a trial space for Ricard's farm. So this is also a place where we can look at changing. He has a 6-block crop rotation. We're not going to suddenly take 10 hectares and be like, let's grow flax. No, he has a plan for his whole farm. But if we take 4 or 5 years as he's working through this new plan to do some things rapidly in the same field, we can get lots of new information that we can think about. How did that function? Did we like it? Did he like it? Do we want to do it or not? So if it's going to work for Ricard, it could work for other people. So the alleys are places where this system, an agroforestry system, silver arable, can give you information that's immediately applicable to other farmers who even don't care about agroforestry. Because it's not just the trees. How? Well, the strip cropping is the pattern, yada yada. I forgot I had so many bullet points on my slide. So there you go. And this picture, the other picture is not mine. This is an intercrop, cereal legume intercrop. Makes a lot of sense. People are working on it. Seems like in Switzerland they're a little bit ahead of the curve in terms of sorting the seeds so that producers can sell it to more than just animal feed. And also, well, there's a whole other webinar about that. In the tree lines, what are we trying to do? See how successional agroforestry improves the profitability, so revenue, crop. It's kind of talked about all this, all right? So this is where we see it going. Polycultures. We just don't have enough data about this stuff. No one's doing it, so we are doing it. That's why. We don't know it works. All right, don't go out and plant your fruit trees 3 meters apart with a rhubarb next to it, 75 centimeters. I heard it from this seminar. I had to look and talk to people and make something that I thought could work. So this is a first stab at this system. I'm interested. If you're not interested, you're not going to do it. So I'm never going to try to argue with somebody who's not interested. You know, if you have an interest, we can talk about your context. And then those trees do bring benefits to the alleys. It is management intensive. Even if it's mechanized, it's more management intensive than planting a field of monocultures. So let's track our time and other inputs and see how it stacks up. You won't be able to say from our numbers whether successional agroforestry works, because think about it, we're not applying fertilizer. Like, come on, it's interesting information, but you won't be able to say the whole concept fails. If we don't make it work, it doesn't mean the whole concept fails. That's what I'm trying to say. And then we're going to manage that diverse community with attention to the target crop. So look at these willows in 1 year. After we got the drip irrigation going, wow, did they shoot off. When we want them to. We want them to grow, and we want to manage their regrowth the same way that farmers manage the regrowth of their cover crops or their pastures through holistic planned grazing. Pollarding, coppicing, in season, out of season, this is holistic planned grazing for trees. Trees will kill themselves if they're allowed to live forever. All right, that's an oxymoron. But what eventually happens is because it has to replace all of the wood every year. Every bit of wood has to be, there's a new layer, keeps going. The productive space can't keep up with the amount of non-productive wood that's being shaded. But if you coppice them, they have a large root system, and then you reduce the burden of reinvigoration every year, and they re-sprout. So oak trees might live naturally 200, 300 years without disturbance. With disturbance, 1,000+ years. And we know this. This is known. This is not, I'm not making these things up. This is known. England in particular. France still has some good observations on that. And what this does is sends these chemicals, signals above and below ground that also the other plants can pick up on. That's, again, on the fringe of what's supported, but it's being done in other climates. And then, again, all of these things are so fascinating. They could be their own webinar, so I can't really talk about it that much, besides to say that here's some pictures of some pollarded trees that were pollarded a bit too late in their life in my neighborhood. This is in Calcolatti. So these are maples. And then there was a poplar. And they're doing it because there's a house. And at some point, a new owner came in and was like, it's too shady, cut my trees. So it would be managed closer to the ground, easier to work. But these things are biological realities. Plants will do this, and you can work with that. And they'll increase their photosynthesis. And then, of course, we're also supporting all of the species with irrigation and foliar applications. A tree to grow old with. So we have an existing working CSA model at Ricard's farm already. People say, I'd like more, I love what this is, can we get some more than apples? So we're on the other side of the farm now because we're not spraying anything, because we don't know how this is going to work, but we're diversifying his offerings to the existing customer base and also people who might become interested. So there's no real change to the actual model. We're just diversifying what's already there. The model works. We don't need to mess with it. Simply that. So what's available each year is what's going to change. And then, of course, we don't have any yield data, so we don't even know how to price the shares to begin with. So we need to work the system in, get the trees growing, see how it goes, and then we help him develop a model that at some point when we hand off management to the farm, that then they'll know, okay, how many kilos do these trees produce? How do I price this? Is this going to work? Do I have to bring the chainsaw out? And then the question about picking, self-picking reduces, and it also improves customer relations, if you want to call it that. People form a bond through these experiences, and if they're willing to do that, we should be willing to work with them if it works for us.
And that's very good for society. That's another benefit.
Yeah, that's one of the objectives for us, is to be working with people, make them interested in agriculture. This is a way to get people interested in agriculture. During the project period, we can't sell anything, or it gets really super complicated if we do. So we're going to be asking that when it's time to harvest, if people want to have some of the yield, that they can take that home if they're willing to collect the data with us, which is not just take everything and dump it on the scale. It's take it from this section, sort it by these different size categories, weigh each size category so we yield map our situation. All right, so there'll be a bit of effort as well, not just take all the garlic and run, but you know, because it'll be a few hundred kilos if we're lucky. And we don't know when we're going to hand the system off. Do I have, oh, I'm running overtime. This is not that long. Hopefully I can go. This is, I usually don't like putting a wall of text, but I want to put this front and center. So successional agroforestry, typically people use that as like syntropic light. They don't want to say they're doing syntropic agriculture, so they say they're doing successional agroforestry. So then you kind of avoid some of the sticky points. But usually syntropic agroforestry and a lot of succession moves towards closed canopy systems. Okay, those have a lot of cons. Okay, there are cons to closing the canopy and creating shade in our situation. We don't have, as you can see, that many crops. There's a whole list of things here. There aren't that many crops we can grow in full shade. It takes a very long time to go there. And by the time you really get a system like that, those trees are so large that if the system's not working, and you have a lot more than 5 tree rows then, they're closely spaced, to remove that situation, to remove those trees, is like removing a forest. So there are a lot of question marks. That doesn't mean that there aren't pros to a closed canopy system. I think we need to investigate these, but you need a lot of land. So when we understand that to support one person's income in Finland, that you need, 60,000 sometimes plus euros of revenue per person to pay them a salary that they want, that means that your systems that you're putting together need to provide that revenue. So you invest in the equipment, in the know-how, the expertise to have these early succession cash crops. And then you're just supposed to at some point say, oh, okay, well, we don't need those anymore. We're switching to these other crops that you haven't heard of that aren't garlic, but they don't taste like garlic, but we don't have garlic anymore. Like, that's not a big selling point for a lot of farmers, unless they have a lot of land that they can, again, then have this shifting cultivation, which I think the world would benefit from, and agriculture would benefit from, but boy, that's a huge shift from the way we see agriculture today, okay? And these systems work really well, though, because they get these biodiverse plant communities to quickly get their roots together. Quickly get the roots, build soil. They're very interesting. I'd love to even start one. But I don't think that they're as broadly applicable in our context. I just don't think that they are. I hope they will be at some point. But I wanted to put that up there. So if you think about our system, we've got these 12-meter alleys, but actually 14 meters between the trees. In a closed canopy scenario, you might even be going, there might be a tree line every 3 meters depending on what the system can support in terms of mulch production. All right, so if it's like, then you're getting the orchard spacing. And now the farmer's thinking about, well, why don't I just plant an orchard? You know, be a lot, especially since we don't have any data to support what you're advocating. So I think that we need trials of these to get started, but I also think that they're not going to be the low-picking fruit. And if you read closed canopy system as food forest, you can also read it as a food forest. Okay. You know, food forests they're very good. There's space for them in the landscape, from homes to farms, but I think that we're a long way away from policy supporting them. So I wanted to say that about the future that I see for temperate successional agroforestry. I think we need these projects to get started, but it's harder to advocate for them than it is for this. But you do lose some benefits like using shade. So we don't get to use shade in our system. So that's why there are no raspberries. If we put a raspberry in that field, we're just gonna have raspberries forever, because we'll never have the shade to get them out, or we'll have to spray them or cultivate them to kingdom come to get them out of there. So we had to reduce our species selection because we don't get the full power of succession. So if we look past the canopy, what else does successional agroforestry have to kind of teach us for our farming systems? Well, soil building can take place. Succession is taking place all the time. It's not just the end goal that you have, it's always occurring. We can look at cash cropping to pay for trees, stratification also for crops. Here's a polyculture, pick your own market garden that I did my last year at Lill-Nägels. All this was planned using the same principles as a stratified perennial system, but I applied them to annuals. This can be done. This is what we're doing. The best cover crops are stratified systems. They're well thought through, niche differentiation, intercrops. So we have a lot to learn. We have the functional diversity that we need to hammer home. You don't need 8 clovers. Maybe you want 2 or 3 nitrogen fixers. You don't need 12 grasses. Maybe you need 3 or 4 grasses at most. Get some functional diversity in the system with some redundancy. And then, the system has the ability to select the most appropriate species. This is something that's really important about those closed canopy scenarios where you put in lots of plants at the same time, and then you allow the site and the conditions to select what's thriving, and then you reorient your management and your decision making according to what is, rather than what you would want it to be. I think that's a really, a very important lesson. So this adaptive management of our farming systems is a huge plus from successional agroforestry. So I'm not bashing successional agroforestry's closed canopy scenarios. I'm saying that we can take a few steps back and allow the really brave pioneers to move in that direction. We're going to kind of work on this for now, and we can work the system and allow it to grow, peel it back, allow it to grow, peel it back, and you can move towards a closed canopy. There's no reason why you can't do it later. I think this is the last one, is it? No, it's not. It's almost last one. Timeline extension, round 2. This is for Stan, our beekeeper. He's asking, what if I'm afraid? I want trees, but I'm kind of afraid to put them in my field. You don't have to put them in your field immediately. You can extend your timeline backwards. So this photograph here is the Viennese countryside. I snapped that when I was leaving this week. Got this. We have some trees already, strips of fields. The farmer's he's out there doing some plowing. Got some good photosynthesis here. And I could draw a nice straight line. You can imagine that instead of putting more trees in these fields, move to something like strip cropping. This is Rickard's farm where we were doing some field trials without trees. So this is what strip cropping can look like. With different kind of cover crops, and this is the cover crop up close. You could probably get paid to put wildlife and pollinator strips in your field, and we know they have advantages. So if you design you like agroforestry, you design your system, but you're not ready to put the trees in the ground, go ahead, put some strips in where the trees would go, begin to shift the soil to a fungally dominated soil, and play it by ear. And if you don't like it, you kill it and you go back to what you want to do. And maybe if you mismanaged it, maybe you have some new weeds. If you didn't mismanage it, you might see, oh, what is that funky nice section there that's growing better than everything else? That's where I had my wildlife strip for 2 or 3 years as I was investigating agroforestry. So you're fighting the weeds before you start implementing the system. It's a low risk scenario. So you're not investing in new trees or new infrastructure. Beetle banks are a Finnish innovation. Maybe someone else came up with it somewhere else. I'll just let you look at the pictures. You get the point. You guys get the point. Here it is, electric powered cultivator. These are both French. This is an Israeli drone thing. But you know, I don't want to sit in the tractor to take care of my, small scale system, but you could have an electric machine that helps you, that assists you in that work, that allows you to scale up without having to bring a tractor out to the field. Battery powered tools already exist for managing trees, shrubs, vines, and everything. They're out there, they're real. These batteries last like 10 to 15 years. So they're also a good investment. And then we won't have time, but yes, drones are coming. And they're going to probably actually allow those closed canopy scenarios to become more realistic, because you could send these fruit picking machines into the canopy. They can go into the canopy and pick, whereas now we wanted the dwarf trees because it's easier to manage. So things are coming, we don't know what yet, but these are all on the radar for farmers who are doing trees and not doing trees. So there's a lot to look forward to. Thank you very much, and hopefully Michelle's not wondering what's going on.
There's 2 minutes before Michelle is on. Michelle is waving there.
He's waving.
He's still happy.
Well, we do have the discussion period, so let me take 1 or 2 questions at most now, and then
Yes, that's fine. In this general audience in the room, is there a question? Yes,
I
please.
just have one on the diversity, because obviously there's the setback of mixing things and making harvesting more difficult, more laborsome. And then, is there a rule of thumb or some kind of distance that would still make it diversified? Like if you had one strip of flax and the other one, what would be the distance where it starts to look like they're not gonna bring the benefits of diversity?
well, it depends on what parameter that you're testing. So we saw some interesting research this week that says that the strips have to be very narrow for some particular outcome. So like 1 meter to 1.5 meters at most.
Yeah.
But really, the 3 meters, 6 meters, even 12 meters is still, remember, better than full-scale monoculture.
Yeah.
So I think there's so little information that we just need people to start
Yeah.
and see what works for them and their equipment, right? So just base it on that and get started with and see where it goes. But we don't really know. And when it comes to the trees, if you're kind of also thinking about the trees, then the low-hanging fruit is to diversify the low strata with mixed perennial vegetation beyond grass and legumes, and not reintroduce all these other woody species. That's the easiest thing that we know we can already suggest, and people are doing it on probably millions of hectares at this point. So, you can diversify without going
Yeah, exactly. What is the... Okay, thanks.
Is there time for one more?